This is lecture 18. In the last lecture, we got into joint compression, focused mainly on the back. And then we talked about injuries very topically. We'll get into that more in the future. And then we spent some time discussing the alteration of neuromuscular recruitment following injury. In this lecture, we'll talk about pain, nociception, and so these are the three topics. Afferent nerve types, not all afferents transmit messages of pain. Some do, and we'll talk about those. Mechanisms of nociception. Nociceptors would be your pain nerves. And then we'll talk about pain chronicity, what gets dysregulated, and how acute pain can transition into chronic. So on the left is an article talking about what pain is. The purpose of pain is to inform you of danger, of some threat, right? So it's for self-preservation. That's why we have pain. But sometimes it can get a little bit haywire. Often, very often, actually, it can. And so we'll talk about how it functions normally and then how it can become abnormal. On the right is a clip I took from a news video from ABC News about the medical mystery of a little girl who feels no pain. It's maybe a little mysterious, but it's this is called CIPA, CIPA, which stands for congenital insensitivity to pain and anhidrosis. So you have an insensitivity to pain with anhidrosis, which means you don't sweat. So here's how this one works. How CIPA works, CIPA. There's a protein we all have called nerve growth factor. And factors are just proteins that do signaling in the way that hormones do. So nerve growth factor, NGF, in the diagram here, is a neurotrophic factor. It's necessary for the growth and the survival of neurons. And this isn't the only factor. A much more widely known neurotrophic factor is BDNF, brain-derived neurotrophic factor, BDNF. And that's similar to NGF. BDNF goes through what's called track B. That's BDNF's receptor. And NGF goes through track A, TRKA. We just pronounce it track A. And that's what you can see on the left side of the diagram. The blue cell surface receptor is track A, and nerve growth factor is binding to it. So when nerve growth factor binds to track A, you get both PI3K, PKB signaling. That's what you see coming off the right side. And you also get MAPK signaling. That's what you see coming off the left side. Although the diagram also acknowledges crosstalk between the pathways downstream from PKB. But in this block, you don't need to know about MAPK or PKB. That's what we'll be talking about in the next block. This is just maybe it's foreshadowing, but also saying how these things work. So what you do need to know, though, is that track A can have mutations. In the left diagram at the bottom, you can see track A, track B, track C, and you can see BDNF binding to track B, but nerve growth factor needs to bind to track A. Tropomyosin receptor kinase A or tyrosine receptor kinase A. Just all you need to know is track A. And if the receptor is mutated, a nerve growth factor won't bind. And that's the primary cause of CIPA. Nerve growth factor binding, the binding of it, will trigger a cell signaling cascade and multiple cascades that are critical to the formation and function and survival of autonomic and sensory neurons. You can also have mutations in NGF, nerve growth factor itself, but the most common, this is really uncommon, but the most common among the uncommon ones is a mutation in the receptor. And tracks A, B, and C, these aren't the only cell surface receptors on nerves. In the diagram on the right, you can see track A, top left of that one. But you can also see a bunch more, and we'll return to this exact image soon before this lecture ends, and we'll walk through all of those other receptors. But again, with SEPA, we lose a couple of nociceptor types, A delta nerves and C nerves. And we'll talk about both of these soon. These are your pain fibers, your nociceptors. But the sweat glands, these lack innervation too, so you don't sweat or you don't itch, but like scratching an itch isn't as important as sweating to maintain your core temperature. So what's most important with SEPA, the sweating is really important, but man, those pain nerves are necessary. So nociception, that is pain. Nociceptors are pain nerves, and um, nociception, right? This is the perception of pain signals. And SEPA isn't the only type of error you can encounter in your nociceptors. Errors that increase pain or just invent pain out of nowhere, that happens too. So lots of stuff can depolarize pain nerves. We'll talk all about that. Um, but lots of stuff can make them function in bizarre ways too, which can cause hyperalgesia and allodynia. We'll return to those terms a couple of times. So because they don't make, I haven't defined them yet, don't worry, we'll talk about those things. So whenever you injure a tissue, you'll get an inflammatory response. Exercise does it. If you've ever felt delayed onset muscle soreness, like after an Alfredson protocol, or just eccentric exercise in the gym, you'll have felt delayed onset muscle soreness. Um, and it's post-damage increases in inflammatory markers. And that inflammation can depolarize nociceptors directly, or it can lower their depolarization threshold. In either case, they're contributing to pain. Now, the nerves themselves will also release substances in the presence of injured tissue. So it's not just the damaged tissue creating an inflammatory environment, the nerves participate too. They release CGRP, calcitonin gene-related peptide, and substance P. Who cares what the P stands for? Nobody actually knows. Powder preparation from the original experiments, maybe. Some articles call it substance pain. But CGRP and substance P are released from the nerves themselves. And substance P, when it loses regulation, we can see this like systemic pain conditions, chronic systemic pain conditions like fibromyalgia, this diffuse and debilitating pain and fatigue. And all you have to do to understand that these people are telling you the truth, they're not just pretending to be in pain, is to look at the cerebrospinal fluid. Tons of substance P in there for fibromyalgia patients. So this is a graph from a study looking at 5 different studies. So each word here is an author's last name, and that corresponds to the study that that author ran. Really high levels of substance P in fibromyalgia patients compared to control. So obviously they're in pain. Only a fool would look at these studies and think fibromyalgia is in people's head, or they're being sissies, or it's not real, or whatever. That's just one explanation of the pain in those situations. So something else substance P will do in addition to nociceptor depolarization is it'll impair motor control. So if you're trying to perform some feat of athleticism, get a PR in some event, or lifting and sprinting, it's whatever, or you have a match or game in some sort of team sport, don't participate while you're sore or in pain if you want to win, right? Show up fresh and painless, or you probably won't perform as well. And it's not just your voluntary actions, your voluntary muscle actions, starting in the primary motor cortex that get affected. Even muscle spindle activity is affected by pain. So let's look at the nerves themselves. There are dendrites to receive messages. That's like the receiving station. There's a cell body, an axon that can be feet long if it's running through the spine. Axons in the brain are those are shorter. But then there's the nerve ending, right? The axon terminal. That's what sends signals to another nerve or maybe to a tissue. And you know that sensory information, that's your afferent information, enters the dorsal root. And then ventral, that's motor, that's efferent. Efferent is motor, that exits the ventral root. So afferent, dorsal, efferent, ventral. And lots of things can depolarize a nociceptor. You can see in the diagram, the nociceptor here, dendrites on the left, right? This is an afferent neuron. It's heading to the dorsal horn, then it sends its messages up to the spine. So you see the dendrites of this nociceptor in proximity to some tissue injury. From that tissue injury, we're seeing bradykinins and prostaglandins and hydrogen ions and ATP, these things being released. This stuff can depolarize those nerves and send messages of pain. So ATP, we know what that is. That's in your cells. That's the energy source of those cells. And it's released from damaged tissue, as you're damaging tissue. Or let's say it's a bladder. Okay, it's a bladder and you haven't peed in a long time and that bladder is distending. ATP will be released from the epithelial cells, the cells that line the surfaces of stuff in your body, organs and blood vessels. Um, a damaged tissue or stretching out a bladder, you're releasing ATP, and that ATP activates the nociceptors. And we'll go through some of this other stuff soon in a little bit more detail. But bradykinins, it's a peptide, it's a protein. Prostaglandins are a lipid made from arachidonic acid. We'll talk about that. Hydrogen ions, that's the H of your pH, so that's acidity. Heat and pressure, that can also depolarize your nerves. But some of these things, in addition to direct excitation of the nerve, they can lower its depolarization threshold. There's a sensitivity of the nerve that can change. And you can also see in the diagram substance P and CGRP inducing vasodilation and activating non-neuronal cells like mast cells, neutrophils. And so this stuff contributes to an even more inflammatory environment. So that's what's called neurogenic inflammation. It's the nerve generated inflammatory augmentation, an increase in the inflammatory environment. One other thing to acknowledge is that under excessive inflammatory conditions or repeated injury, chemical receptors can be made more sensitive and increase in number. And the more receptors you have, the more binding you'll experience. And the more binding causes more triggering of the nerve. So that makes the nerve more responsive to the same concentration of chemicals. So if this is a nociceptor we're talking about, you'll feel more pain in response to the same chemical concentration. Okay, so we have our nerve ending here. And remember, we've talked about GABA and glutamate before. GABA is the primary inhibitory neurotransmitter in the central nervous system, and glutamate is the primary, most abundant, excitatory neurotransmitter. There's also glycine. We've talked about in the context of the Golgi tendon organ, the inhibitory interneuron in the spine that hyperpolarizes the alpha motor neurons. Those use glycine to do it. And glycine is it's a main inhibitory neurotransmitter for the interneurons. And it's involved in some brain areas that we don't need to talk about related to like sensory motor processing. But glutamate Glutamate is the primary excitatory neurotransmitter in the central nervous system, and that's what we're looking at. A nerve terminal is releasing glutamate to depolarize another nerve. And there are lots of receptors on the surfaces of our neurons. So here's the presynaptic neuron releasing its glutamate, and that glutamate can bind to ionotropic receptors or metabotropic receptors. But let's start with the ionotropic receptors. So what do these things do? Let in ions, right? Ionotropic. They let in ions. How do you depolarize a nerve? Ions. Earlier in the semester, we talked about action potentials being caused by the influx of sodium. That's an ion. So that's what ionotropic receptors are doing. They're letting in ions. Among these ion channels, there's AMPA, and you absolutely do not need to know what it stands for, but this you do need to know what it lets in, and it lets in it's the influx of sodium that it's permitting. It brings a positively charged ion into the cell, and so it'll depolarize. Now, NMDA receptors let in both sodium and calcium, and calcium is more regulatory. Pain medications like gabapentin for neuropathic pain exert some of the effect by antagonizing the NMDA receptors. So long-term potentiation, that means like more robust excitation in the future, that requires NMDA activation. Again, calcium has regulatory roles. And the same is true for long-term depression, meaning a less robust excitation in the future. NMDA receptors help regulate the AMPA receptor expression. Now, who cares about the kainate receptors? For the sake of this class, let's ignore them. Not who cares, but it's another ionotropic receptor, and these postsynaptic the kainate receptors. What we're looking at here, these are involved in excitatory neurotransmission, but they're just not very well understood. So we're going to ignore them. Um, and as you continue to activate a nociceptor, activate it and activate it and activate it, you can change the structure and behavior of those receptors. You can upregulate the number of the receptors present. So, like pain propagates pain, pain facilitates future pain. We're really good at coping with things like DOMS, delayed onset muscle soreness, without negative consequence. So don't worry about delayed onset muscle soreness. Our ancestors' lives were just nothing but DOMS. I mean, since like time immemorial, DOMS. I mean, track your ancestors back, and they were just beating up their bodies day in and day out. But more than DOMS, beyond DOMS, actual injuries, we're starting to get into some plastic territory. Plasticity means like shapeable, moldable, malleable, alterable. Plasticity, we can change things. But if you've injured yourself before or had some other source of lingering pain, your depolarization threshold might be lower. And if you have more postsynaptic receptors, you'll be more sensitive to the same dose of chemicals, of neurotransmitters. The same signal might give you a more pronounced effect. Okay, the metabotropic receptors, these work through second messenger systems. This just means a signaling cascade. In this case, it's PKA signaling, protein kinase A. It's not just letting ions in. The ionotropic receptors have an ion channel 4. The relationship between metabotropic receptors and ion channels is indirect. There's a signal transduction pathway, again, just cell signaling. And those signaling cascades have diverse effects. The metabotropic receptors seem to modulate the other receptors. And we'll look into that specific signaling in just a minute. But there's a expression, there's a cliche that genes that fire together, wire together, really anything that fires, nerves fire together, wire together, everything that fires together, fires together. I'm like destroying the cliche here, um, but that's plasticity. There's um, the way metabotropic signaling is regulating these things. There's a lot more genetic expression differences. So we've talked about this before, but lots of things will depolarize the nociceptors to cause an action potential in a pain nerve, right? Histamine, bradykinins, ATP, hydrogen ions, stuff like that. Now we've seen this top left picture before briefly, some 15 minutes ago or something like that. The fifth slide, I think it was in this lecture. So we're looking at the exact same thing. The cell surface receptors on a nociceptor. Now, in these diagrams, in each of these diagrams, it's just showing one of each receptor. And that's not what a cell surface looks like. It's loaded with uncountable receptors. And increasing the number of receptors will almost always lead to increased responsiveness, and vice versa. Decreasing them or inhibiting them will almost always lead to a decreased responsiveness. Okay, so let's go through this. Um, our nociceptor that has a bunch of cell surface receptors on it. First up, TrkA, tropomyosin receptor kinase A. Nerve growth factor binds to it. If you aren't initiating this signaling cascade, you have CEPA, right? Congenital insensitivity to pain with anhidrosis. Because nerve growth factor is necessary for the maintenance and survival and proliferation of neurons. Without it, apoptosis will happen. So just revisiting CEPA, CIPA, um, tropomyosin receptor kinase A or tyrosine receptor kinase A, mutations. That's the primary cause of this. The gene that makes that protein is making a defective version of it. And without it, you'll wind up with an absence of C fibers and like a drastic reduction in your A delta fibers, maybe an absence of those two. So an inability to sense heat and sense pain. I mean, the same nerves that should be transmitting pain sensation should be transmitting heat sensation. So if you don't transmit any of that information, your brain doesn't know how hot or how cold your body is. So thermoregulation gets compromised too. Over here, we see a pore through which sodium is coming in. This should remind you of an AMPA receptor, an ionotropic receptor that lets sodium in. Over here, you see calcium and sodium coming in. So that's an NMDA receptor. Remember, both ions flow through NMDA, ionotropic receptors, and the calcium flux is important for the ionotropic plasticity, long-term potentiation and depression. If you see hypofunction of NMDA receptors, synaptic plasticity will be impaired, which does have implications for learning and memory, not just pain transmission. And again, lots of drugs will affect these. Gabapentin is a non-competitive antagonist of NMDA receptors. We'll talk about what that means in the next block. Non-competitive inhibition, that's a next block topic. But drugs target these. And here we have bradykinins. A bradykinin, it's a peptide, or it's a string of amino acids that causes vasodilation and it amplifies other processes while depolarizing your pain fibers. So bradykinins, that's another target of drugs. I mean, one thing aloe vera has in it is a bradykinase, which breaks down bradykinins. It won't be all that helpful for delayed onset muscle soreness, but it'll like help with your sunburn, and that's not fake. But opposite that, ACE inhibitors inhibit the breakdown. Of your bradykinins. So levels will increase. And since bradykinins are vasodilators, blood pressure gets lowered. So in addition, like the amplification effect of bradykinins, in addition to all that stuff, they're 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 augmenting AMPA and NMDA receptor activity. So that's another form of amplification of pain sensation, increasing the AMPA and NMDA activity. And then also inside, they're releasing prostaglandins, bradykinin are releasing prostaglandins intracellularly. And I'll talk about prostaglandins in a minute, where they come from, how you get them. But the EP you see in the diagram, that's a type of prostaglandin receptor, as opposed to DP or FP. And B2, which you also see, that's a bradykinin receptor. So bradykinins do a lot, but keep prostaglandins fresh in your mind, because we'll talk about them in just a minute. Down here, you see a receptor for CB or opiate. CB stands for cannabinoids or cannabinoids, however you want to say it. These technically aren't the same receptor. Your cannabinoid receptors and your opiate receptors, totally different. But the signaling cascade triggered, PKA signaling, protein kinase A signaling, is the same. So sometimes people will lump these together, cannabinoid and opiate uh, receptors together in the diagrams, despite being totally different drugs with totally different constellations of effects. But there's some overlap with them. And so let's talk about the overlapping effect in this particular transduction path. How would marijuana or morphine cause pain relief? By inhibiting adenylate cyclase. And um, adenylate cyclase, or sometimes it's called adenylyl cyclase, is an enzyme, and the ACE is an enzyme, and this one converts ATP into cyclic AMP. Um, Cyclic AMP binds to PKA, which is protein kinase A, and PKA phosphorylates things. What kinases do is attach phosphates to other molecules. Protein kinase A, PKA, being a kinase, attaches phosphates to things. So in this case, it's attaching phosphates to things that would help induce pain and remodel a nerve to be more like pain oriented. So again, how this cascade works is, if you turn on adenylate cyclase converts ATP into cyclic AMP. Cyclic AMP binds to PKA, that activates PKA, and then PKA conducts its signaling cascade. Um, Something you see binding there is AEA. That's your body's endogenous cannabinoid. It binds to those receptors. It's named after the Sanskrit word anandamide. It's the Sanskrit word for bliss. Now, opposite, weed and morphine is prostaglandins. I mean, literally opposite in this particular case. These are activating adenylate cyclase, where previously we were talking about inhibitors of adenylate cyclase. These are activating it. So they're increasing the conversion of ATP into cyclic AMP, and in doing so, increasing the amount of phosphorylation that PKA is doing. So prostaglandins will activate this PKA cascade. And prostaglandins, this is the bulk of your delayed onset muscle soreness and your hangover. If you're going to like exercise while drunk, something I hinted at last lecture, you might have a lot of prostaglandins to deal with. So how NSAIDs work, ibuprofen, stuff like this, how these work is by inhibiting the enzyme that converts arachidonic acid into prostaglandins, and that's cyclooxygenase, or COX, COX, cyclooxygenase. And so you reduce the amount of prostaglandins you have if you inhibit cyclooxygenase, block your COX, right? You reduce the amount of prostaglandins that you have, and less prostaglandins then are binding to the receptors. And so that's almost like taking morphine, in this one very narrow way. Each drug has a lot of other effects in addition to this. But in this pain pathway, that's how NSAIDs work. And they're non-steroidal anti-inflammatory drugs. NSAID, non-steroidal anti-inflammatory drugs. An even more effective way of inhibiting the prostaglandin signal is through SEDS, right? SAIDS. No one really calls it that, but that's what it is. It's steroidal anti-inflammatory drugs. A cortisone injection um, is a steroidal anti-inflammatory drug. Um, so like NSAIDs or Aleve and Advil, those are non-steroidal, and cortisone is a corticosteroid. And cortisone is one step away, one enzymatic step away from cortisol, a steroid hormone that's made in your adrenal cortex. Hydrocortisone, the drug, is cortisol. That's just what cortisol is called when it's used as a medication, as hydrocortisone. And what this does is block an enzyme called PLA2, phospholipase A2, PLA2. And PLA2 releases arachidonic acid from the phospholipid bilayer, from the membrane. And I'll talk about this in just a minute. But if you release the arachidonic acid, then cyclooxygenase, COX, can convert it into prostaglandins. But for cyclooxygenase to work, it has to have access to the arachidonic acid. So PLA2 is what gives COX its access. So a cortisone injection is very anti-inflammatory. All right, so let's zoom out and look at a neuron with even more stuff on it, more cell surface receptors. So that's what this is. The gray rectangle, that's your phospholipid bilayer. And outside of the gray is the outside of the nerve. And inside the gray is inside the nerve. Now it looks, really busy, but if we eliminate some of that busyness, we'll see a lot of things that you recognize. Top left, you see bradykinin receptors. You know what that is. You see prostaglandin receptor. You know what that is. At the bottom left, you see a couple of different prostaglandin receptors and CGRP. Remember, that's something the nerve releases. It's part of neurogenic inflammation. We see overlap with the mechanism of prostaglandins and CGRP on PKA signaling. We're activating adenylate cyclase. That's what the AC is. Adenylate cyclase converts ATP into cyclic AMP. You see that. Cyclic AMP binds to PKA. That's what activates PKA. And now we're getting PKA signaling. And then at the bottom, you see the opioid receptors. And GABA receptor. You know what that is. You know what GABA is. Gamma aminobutyric acid. And you see the cannabinoid or cannabinoid receptors. Cannabinoid 1 and 2, THC is great at binding to the CB1 receptors. You also see AMPA and NMDA. And then on the right, you see GABA and glycine, ATP, heat, and proton, which just means hydrogen ions. So a lot of this stuff you should recognize. But prostaglandins, okay, where did these things come from? Arachidonic acid is one of the lipids in your phospholipid bilayer. It's a 20 carbon polyunsaturated fatty acid, and it just lives in those membranes. So, like in skeletal muscle, for the membrane of skeletal muscle, the arachidonic acid content is pretty high. Maybe it's 10 to 20% of the total fatty acids there are arachidonic acid. And exercise damages the tissue a little bit. And then phospholipase A2, that enzyme releases your arachidonic acid. That's the enzyme that a cortisone injection would be inhibiting. It releases phospholipase A2 releases arachidonic acid. Then COX, cyclooxygenase, converts it to prostaglandins. The name sounds like prostate gland. That's because it's what it's named after. The compound was identified in the 30s, 1930s in the prostate, but they're everywhere. I mean, prostaglandins are made by like everything. Any nucleated cell is going to release arachidonic acid. So we've been talking about pain nerves all lecture long, but remember we have efferent nerves too. We have motor nerves, and things interact with those nerves too. So I just want to take a moment to look at the bigger picture of nerves in general with a little story. You don't need to know the exact story, but there's some pieces here, some physiology to know. So imagine it's the 1820s outside. It's a century ago. Some Germans just ate some improperly prepared blood sausages and they died. And then Justinus Kerner looks kind of like a shepherd, right? He wasn't. Studied those sausages and failed to solve the mystery. Instead, he explained that there was just it was a worst gift, right? Which is German for sausage poison. So inside of the sausages, there's a worst gift, WURST. So think like bratwurst. WURST, except in this case, it's sausage poison. It's worst gift. So that's what he came up with. And then he ran some experiments, Kerner did. And with the tiniest dose, he found muscle weakness. And the larger the dose, the more paralysis he was finding. And it was pretty easy to kill critters with it. It would be easy to kill people with it too, as evidenced by the sausage. Um, but in the muscle weakness range, in that dose, he was finding things like droopy eyelids. And we've talked before in this semester about a cause of droopy eyelids, and that was myasthenia gravis. It's an autoimmune disorder where you have antibodies that eliminate the nicotinic acetylcholine receptors. And you remember how those work. At the very end of an alpha motor neuron, when the action potential has to cross the neuromuscular junction, it does so with acetylcholine. And that acetylcholine has to bind to nicotinic receptors. So Kerner found similar complications with a tiny dose of sausage poison. So you should have an idea of the physiological pathway that's causing the droopy eyelids. So Kerner dies. Um, he doesn't figure it out how it works. But then some Belgians ate bad ham, improperly cooked ham, and the same thing happened. People die. Some people die. Other people don't quite die, but come close with paralysis and so the meat is sent to the University of Ghent, where this well-mustached professor studies it. And he looks at the little culprits under a microscope, and wouldn't you know it, they look just like sausages. I mean, Kerner was right. It really is sausage poison. So he calls it the same thing as Kerner did. He calls it literally sausage poison. He just used the Latin for sausage, botulinus. Bacillus, it means a little wand or shaft or like staff. So Bacillus botulinus means like little shaft of sausage. And then botulism would be sausage disease. So botulinum toxin is what we call it today. But now we're including Greek because that toxin, that's an archery term from like a warfare history of dipping arrows in poison. So like detox literally means elimination of archery. But that was right at the end of the 19th century, the 1890s. Fast forward the world a century, get into the 1990s, and sausage poison is just rampant in Hollywood and elsewhere, but on purpose now. It's on purpose. So what is it, right? What is sausage poison? What is this compound that Americans, especially today, I mean, just Americans alone are spending, $2 billion every year on sausage poison. So what is it? Botox, right? Botulinum toxin, type A specifically, it prevents the release of acetylcholine from your motor nerves. Um, so, maybe if you get the, botulinum dose just right and combine it with atropine, you could cure sarin gas poisoning, right? Nerve gas, but I wouldn't try, right? Neither would I be surprised if it were like the next Tide Pod challenge and it becomes like the leading cause of death in like social media users. Um, but what you should know from this is not the history part, but that stuff affects nerve transmission, afferent and efferent. And in the case of efferent, in the case of motor, know these things, right? Myasthenia gravis, an autoimmune condition. And so that's not like a drug you take, but this condition destroys the nicotinic acetylcholine receptors. Sarin gas, your nerve gas, that blocks acetylcholine esterase, which, if blocked, you can't break down acetylcholine, so you can't stop transmission. And then atropine or atacurium, these are examples of anticholinergic drugs. You don't need to know the specific names of the drugs, but anticholinergic drugs. These block the binding of acetylcholine to the receptors. So atropine inhibits muscarinic receptors and atacurium inhibits nicotinic. Again, you don't need to know which drugs target which receptor. Just know anticholinergic drugs. And then there's Botox. You know how this works, preventing the release of acetylcholine from your motor nerve. So know those four things: myasthenia gravis, nerve gas, anticholinergic drugs, and Botox. Now, how does the haywiring happen in our once we have pain? How does acute pain become chronic pain? Substance P is one way. Dysregulation of substance P leads to chronic pain, which serves no useful purpose. It's not warning us of behavioral or environmental threats. It's sounding false alarms, like the boy who cried wolf, except it's nerves crying pain when there's no wolf bite at all or anything else that warrants the crying. CGRP2, if this loses regulation, just like substance P, it'll contribute to a chronic inflammatory environment and pain chronicity. There's a term that's used in this study here, central sensitization. We'll define that later in this lecture. For now, just think of it as the nociceptor system crying wolf every time it sees a picture of a puppy. And then also the PKA signaling we were talking about. Remember, prostaglandins activate the PKA pathway in nociceptors, and this is inhibited by cannabinoids or opiates. Opioids are just that just refers to synthetic versions of natural opiates. So if you see opiates or opioids, just lump these in. It's the same thing, natural versus synthetic. Um, so if PKA signaling, if that's not well regulated, this can contribute to neuropathic and inflammatory chronic pain too. So the terms I've been referring to, central sensitization, that's one of them. Your afferents are overexcitable in the central nervous system, central sensitization. And there are lots of causes of this. Nobody knows them all, but the consequence is well known. Pain signals get amplified. Now, there's a difference between hyperalgesia and allodynia. Hyperalgesia is a hypersensitivity to pain. Whatever the pain is, it's exacerbated. Where allodynia is it's a not just it's a painful response to non-painful stimuli. Should be innocuous movements, proprioceptive things moving around, mechanical sensation. That instead gets transmitted as pain. So we have different afferent nerve types. There's A alpha and A beta, thickly myelinated nerves. These aren't sensing pain. These aren't nociceptors. A delta and C, thinly myelinated and unmyelinated, these are the ones sensing pain. And the type of pain you experience from them is different. A bee sting or a tattoo pen, that stuff is sharp. You know exactly where the bee stung you. I mean, you can close your eyes and point to the stinger. That's A delta fibers. C fibers are much duller, they're achier, they're more diffuse. If you have a stomach ache, where exactly does it hurt? You just sort of in your all overs. If you have some deep muscle ache, it's not precise. You just rub an area and say, like, oh, it hurts all over here. That's a bunch of C fiber transmission. The bee sting is A delta transmission, sharp, quickly arrives, quickly resolves. C fibers are slower to set in. They're diffuse, but they're slower also to dissipate. Now you don't need to know this, but nerves aren't isolationists. They interact. There's communication between nociceptor types. One mechanism of this is that C fibers can release CGRP, and that can bind to the A delta fibers. So A alpha, A beta, these things move fast, like speeding Ferraris, top speeding, cars. Nerve conduction with these is very, very fast. Um, but think about what we do when we injure ourselves. Your hand gets stepped on by a cleat, like hurts, whatever. That's um, tons of A delta fibers firing. We shake it. You shake your hand. Yeah, you're kicked by a cleat. You like rub it, right? This is a bunch of A delta activity and rubbing the injury, shaking it. That's not helping the healing, but it is relieving pain. That's why we just, as a species, we do this because you're activating the A alphas and the A betas, really thick myelination in those A alphas and A betas, really fast nerve conduction velocity, and they're sensing mechanical stimuli. Um, they're sensing things like the pressure of rubbing tissue or the proprioceptive forces when the tissue is being shaken. That's what they're sensing. And your brain is not a very good multitasker. Read a book silently while having a conversation with someone and like while trying to solve a math problem in your head. You can't do that because you can't pay attention to those things at the same time. We can like walk and chew gum. That's not multitasking. Um, just like you can walk and make new red blood cells and, convert arachidonic acid into prostaglandins, you can do all that at the same time. That's not multitasking because that's not really using your brain. Even the walking is just coming from central pattern generators in your spine. The brain isn't good at real multitasking. And so stimulating bigger, thicker, faster afferents can distract the brain from processing the A delta firing that's coming from the cleat wound. So that's why we're shaking and rubbing our A delta injuries. Um, and when we're looking at allodynia especially, we can see a rewiring of those A beta fibers though. So sometimes the A beta fibers can participate in pain. They're not supposed to. So if you're playing that game of telephone, like when you're a little kid, I don't know if you've done this, everyone like sits in a circle and you whisper something to the person next to you, and then they whisper that same message to the person next to them, and they whisper to the next person, and so on. And then it gets back to the original whisperer, and the message is always distorted a little bit. That can happen with nerves too. The message that is transmitted from the periphery can be heard as something else. That's not what I said. I sent a message about proprioception, shaking or rubbing or whatever movement. I didn't say ouch, but that's the ouch is the message that ends up getting interpreted. So that's a rewiring at the dorsal root. Getting to the A delta fibers, again, these are the ones that have a little bit of myelin. They have some myelin, not as much. They aren't as fast as the A alphas and A betas. They're, world-class sprinters, like Usain Bolt and whatever. That's how fast the nerve conduction velocity is in these. And so think about like icy hot and acupuncture and pinching yourself for a headache. And part of what each of these is doing, not everything that each one is doing, but part of what they're doing is stimulating the A delta fibers, which are thicker and faster than the C fibers. So make your brain pay attention to those inputs, and it might ignore the alternative pain inputs, the C fiber input. So getting into the C fibers, these are slow. They're a casual walk. That's their nerve conduction velocity. It's you walking between classes, like assuming you're not late. And again, what you're feeling is like achy. It's not quick and sharp pain like the A deltas. So taken together, the A deltas and the C fibers, these are your nociceptors, your pain fibers, and they respond to all of the stuff we've been talking about, heat and pressure and chemicals, but they're also silent nociceptors. Once these are awoken, people often experience um, more of a chronic, but like after surgery, for example, people will experience a chronic pain. The surgery awakens these nociceptors. They just fire and they continue to contribute um, to pain in a pointless way. You're not still having the surgery a year later, but those nerves are still firing. So you don't want to wake up rather the sleeping nociceptors. Um, just like you wouldn't want to wake up a sleeping baby if you know it's going to scream, it's going to shriek, right? Just let them slumber, and let's keep things quiet. So this is just a look at the three different classes of afferents, A alpha and A beta. Remember, these aren't nociceptors. They're sensing mechanical stimuli, light touch, non-painful pressure, proprioception, that sort of thing. But looking at the two nociceptor types, they're the A deltas and the Cs. We see the A deltas being the initial pain, right? That like the first responders in a sense, sounding an immediate sort of extreme alarm, but they don't usually linger in the way that the C fibers do. C fibers are the ones that really nag at us. Um, and when you look at the diagrams to the right, this has nothing to do with the action potential of the nerves. I just wanted to make sure that's understood. Um, the action potential is the same. The A delta and C fibers don't have different, versions of action potentials. They just have different behaviors upon activation. Now, where do these nociceptors live? The A delta and C fibers are both in superficial organs, think skin, right? But in the deeper structures, your muscles and your joints, that's mostly C fibers, which is why a pinprick will have a sharper sensation. But in the muscles and joints, your sensation tends to be duller, and we have poor localization of it. We can't like have a pinpoint of exactly where we're feeling that pain. So we've talked about these a little bit, right? Hyperalgesia and allodynia. But I want you to remember is that hyperalgesia is an amplified pain response, and allodynia is sensation that should not be interpreted or felt as pain, but pain is the feeling you get. Pain experienced from non-painful stimuli. That is allodynia. Hyperalgesia, uh, you should be having a little bit of pain, but why is it this painful? And then allodynia, like this shouldn't be painful at all. Why am I experiencing pain? And then there's peripheral versus central sensitization. This means we're being sensitized to pain either in the periphery or central at the dorsal horn. Pain chronicity can occur in either location. In the periphery, there's generally an inflammatory environment that alters the depolarization threshold of the nerves. Then at the dorsal root, it's more of a misinterpretation of signals. And remember, afferents affect efferents. So chronic pain is not something that athletes want to subject themselves to, or anybody hoping to produce good, feats of athleticism, human performance. Pain is an opponent of that. So that's it. But our review questions are abundant for this one. So there we have 4 slides of review questions. Know about nociceptors. Know the difference between GABA and glutamate. And know some things that depolarize the nociceptors. Know what neurogenic inflammation is. And within that, what substance P and CGRP are, what are those things? Make sure you know the difference between ionotropic and metabotropic glutamate receptors. Within ionotropic, know the difference between AMPA and NMDA receptors. Know about long-term potentiation and depression, and then know just a few major steps in PKA signaling. That's going to be a bigger topic in the next block. And then these, CIPA, CIPA, track A, ATP in the bladder, we talked about that. The relationship between arachidonic acid and prostaglandins and how NSAIDs and steroidal anti-inflammatories work. And know about the types of nociceptors and the difference between opiates and cannabinoids or cannabinoids on them. And then know how Botox, anticholinergic drugs, sarin gas, and myasthenia gravis, how those work, the mechanisms for each, again, briefly, but the mechanisms for each. The difference between allodynia and hyperalgesia, the difference between peripheral and central sensitization, what explains delayed onset muscle soreness, what bradykinins do, and what explains the transition from acute to chronic pain. And then that's it. That's a lot, but that's all of the lot of it. And I will see you in lecture 19 when we get into injuries.